Hi class, so today we are going to talk about the big reveal, the summary of what we have been trying to accomplish in this DNA forensics project for the past 4 weeks. So I'm going to talk a little bit about the background of the assignment. I'm going to talk about some of the reading resources that you began reading in week 1 and hopefully continued to revisit over the course of the unit. And then I will tell you what the big solution is to the identity of this mystery organism. So in our assignment, you were told that some genetic material was found recently on a meteorite that had impacted with the Earth. And the media became very excited and said this is the first evidence of alien life. And moreover, this life is very similar to life on Earth because it has DNA and some very, very similar DNA to what you would see on Earth. And your team, a team of evolutionary biologists, was called in to verify these claims. And understandably, you were skeptical that this was evidence of alien life. The odds of that are fairly astronomical. And so an example of a hypothesis that would fit well with the project that we've been working on is if the genetic material is of terrestrial origin, if it's from this Earth, then it will share significant sequence identity with a known terrestrial organism. So you basically said that if you were to compare the DNA to other known organisms, you would find large similarities. And I think that's a pretty sound hypothesis because you would not expect an alien organism to share a lot of identity with one from Earth. These genes have all evolved together and so should share a great deal of identity with one another. So just moving into a description of what we've been doing, we have been basing our work on this principle that species relationships can be determined based on how similar their DNA or protein sequences are to one another. And part of the reason for that is that through evolution, organisms have diverged from common ancestors. So originally, at the onset of life on Earth, there was an organism that was some type of prokaryote. And a modern-day prokaryote that we know about today would be E. coli. And then over time, the prokaryotes, some of them, a subset of the prokaryotes, developed some eukaryotic traits. They developed organelles, and we'll talk more about these eukaryotic traits later on in the class. And these eukaryotes, one representative of that we've talked about in this unit is yeast. And then all of these other organisms are examples of eukaryotes. So over time, the eukaryotes would have diverged into different types of organisms. The C. elegans is a type of roundworm. Drosophila is a type of arthropod. These would have diverged early. These are the invertebrates. And then later, we would have also seen the vertebrates diverging. And so we would get things with internal skeletal structures, such as fish. The zebrafish is an example that's commonly used in research. Frogs, so Xenopus, we talked about in class. Chickens, so birds. Mice would be an example of a mammal, and humans as well are an example of a mammal. And so the most closely related organisms in this image would be the human and the mouse, and then more distantly related organisms are separated at these branch points. And we'll talk more about this as well over the course of Unit 3. And so the organisms that we looked at were covering a range of evolutionary distance from more ancestral characteristics, so those that share characteristics with organisms that would have diverged very early from the first living organism on Earth, and then progressively, more recently evolved organisms. And what we see is that the more distantly related a pair of organisms are, the more time that has been available for differences to accumulate within their genetic structure. So all organisms share genetic identity for certain genes, and we'll talk more about that in a minute. More recently diverged organisms may have genes that do not appear at all in the more ancestral organisms, and part of that is because they've developed some traits that are not possessed by these more ancestral organisms. And so we've seen examples of that as well. Protein structure also changes as a consequence of those changes in the DNA, and the amount of change that you'll observe in the protein structure between organisms over evolutionary time is going to vary by the protein. And so for types of proteins that need to be very conserved evolutionarily because their function is very basic to life, like in this example they're showing histones. That's a very basic function to life, and these proteins have to have a very specific structure in order to perform that function. And so there's not much room either in the protein's shape or in its function for variation to occur. Other types of proteins may have more propensity for variation in their shape, in their purposes, and they may have more room for variation because the purpose, the function that they serve in the organism may not exist in certain types of organisms. When you're working with very distantly related organisms, you want to use amino acid sequence similarity over DNA sequence similarity, and that's largely because of this concept that we've talked about multiple times in class, this concept of the degeneracy of the genetic code. And so, since there are cases in which you could have multiple codons encoding the same amino acid, then the rate at which DNA is going to vary is going to be much higher than the rate at which amino acids are going to vary. So you'll see change in amino acid sequence more slowly over evolutionary time than you will see change in DNA sequences over evolutionary time. And so if you're comparing very similar species, you're going to want to use DNA because that is going to amplify the differences between those organisms. With the amino acid sequences, this is very useful for very different organisms because you won't get as much noise from those silent mutations. You can look more deeply into this process if you would like. You can look at examples from other research, and these are just a couple. So some examples of that would be hominid evolution. So they're looking here at differences in DNA structure. There's actually 98 to 99% similarity between humans and other types of primates. So just a very small amount of genetic diversity can create a large effect on the final structures and functions of the entire organism. And then for things like the evolution of life on Earth, you would want to make sure that you're using amino acid sequence, again, because you're covering this huge amount of evolutionary time. So to get down to what is our mystery sequence, let's talk a little bit about some of the variation that we've seen in this project. So a couple of the genes that we used in our homework unit would be G6PD, and as you guys noted in your resources, G6PD is involved in the pentose phosphate pathway, and it is important in the manufacturing of NADPH in the cell. And if you think back to 151, you might remember that NADPH is critical to the metabolic pathways of all types of cells. And so it's necessary for the generation of ATP. And so it shouldn't surprise you very much that this gene is present across this vast amount of evolutionary time. From the most ancestral organism that we looked at, these prokaryotes, to the most recently diverged organism that we looked at, the mammal. And it should also not surprise you that there's a pretty high amount of conservation, and so there's a difference between the mouse and the frog that we looked at for this gene, and there's not that much difference when you go back to the arthropods, to the fruit fly, and then again the difference is not that dramatic when we go back to yeast. It's quite different from our original organism, but this gene is fairly conserved over evolutionary time, and that's because it performs a very critical function, and you don't want to lose this function. And then we also looked at some genes important to some more recently diverged organisms. And so 6.3, as you guys also noted in your resources, is a gene that encodes a transcription factor. And this transcription factor is responsible for organizing the development of eyes and brain structures. And so it is absolutely useless to organisms that don't have those organs. And so it is not found at all in E. coli, the prokaryotes, or yeast, that one of the earliest forms of eukaryotes. It is found in Drosophila, and then it is quite similar between the frog and this mammal, so this amphibian and this mammal. And similarly, it should not be so surprising that such a recently diverged gene should have less conservation than one that is more fundamental to an organism's survival. And as I think the entire class was able to notice, the genetic similarities for the frog were the highest for this project, but they were not completely identical to one another across different types of genes. And so the mystery DNA was absolutely identical to the frog for more fundamental genes like G6PD, and then for more recently diverged genes, the similarity was reduced. And there was some debate in the discussion forum about why that might be, and some people hypothesized that perhaps there was some degradation of the mystery DNA, and that's a good hypothesis. But you would expect that there would be a similar amount of degradation across the DNA as a whole, if that were the case. So you should see, about 95 to 98% similarity across all the genes, if this was still from Xenophus laevis and the differences were due to degradation. And those would actually be detectable as you analyze the DNA, as you sequenced the DNA. Your machinery would be able to inform you that you were not sequencing DNA that had high integrity. Some other individuals hypothesized that perhaps it was the mystery organism was still a frog, but maybe a different species of frog. And that is exactly what has happened here. And so the frog that we collected our mystery DNA from was this Xenophus tropicalis. And so it is a fairly close relative of Xenophus laevis. They're both used in laboratory settings, and both have genomes that are not entirely, but almost entirely sequenced. And so you can see some of the similarities and differences between them in this chart here. But as you noted in the homework unit, their genetic similarities for these genes are quite high. And I would like to congratulate you all on your hard work in this homework unit. I think that it's been a really interesting journey, and I've really appreciated the time that you've put into making this project a success. I am looking forward to reading your reflection journals, so go ahead and make sure that you write up a paragraph or two this evening about some of the things that you've learned in this unit that maybe you found surprising or that you'd like to know more about, or to talk about some of the strengths and limitations that you see in the techniques that you've learned. And of course, this journal entry is private between you and me, and I would like to hear as well any thoughts that you have on improving this homework unit for future students.